We already started, but we didn't get very far.
Previously on Biology 1070, we just blathered on. But now there's a quiz. So shh, let's watch.
That was a really good reminder.
Oh, yeah. We would have, like, blah, 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 blah. We'd end up complimenting each other. That was the best way that's ever been explained.
Okay. Welcome, and what we're going to do is shut it down in just a few more seconds. So.
No, wait, Riley just got here.
Oh, Riley's just coming in. Hi, Riley!
Everybody say, hello, Riley!
Hello, Riley! No, sorry.
Sorry.
Just...
What have I missed?
So sorry. No, everything's fine. We're just doing a poll, and I will be shutting it down in about 10 seconds.
One Madawaska, two Madawaska.
No, it's done. It's done. It's over. There are no new participants. We almost hit 80%.
I know, you were holding up for 90 last time.
I know. Wishful thinking. So there you go. Sharing the answer with you. The answer is down here. It is the one that is not. Thank you to whoever circled it right there. That is the one that is mismatched. Well done. So go ahead and go through them again now that you know that the answer is E. We're looking for the one that is properly matched. So now you know the answer is E, and you can work through it. Obviously, don't just like find the answer, but also try to explain why the other ones are not the answer, or rewrite, even better, rewrite the statement so that it does match. Those are two ways that you can use these types of questions. Okay.
I just maximized Scout's window, so I'm like, I want to go like...
Oh, yeah, we're all just like watching Scout. Yay! Hi, Scout. We should give Scout a credit. Okay. Why don't fish freeze? There's no segue to this. No. Okay. So there's a nice little thing to kind of like think about for the next little while. And that has to do with...
Flounder?
Flounder? No.
Build up the momentum again.
Yeah,
Here we go.
the winter flounder. Okay, so a nice little kind of puzzle and a nice little story about science that was done, and we're going to try to like integrate you into this type of thinking. Okay, so you have a fish that lives in really cold water, a winter flounder, and the water, seawater, freezes at -1.9, not at 0. And the temperature of seawater can get to -1.9 before it turns into sea ice. So we have fish living in these places in the Arctic and the Antarctic that are living in temperatures that are below the freezing point of fresh water. However, they are regulators of salt, and their bodies are made of fresh water, not salt water. So freshwater freezes at 0 degrees. How is it then that a freshwater fish, bodied fish, can live in a saltwater environment that is below the freezing temperature of its freshwater body? It's a nice little puzzle. And so there are many, many, many possible answers. And the delightful thing about, about nature and biology and biodiversity is that there are maybe as many answers as there are species that have solved it, right? So it but like a lot.
No, I'm just trying to come up with Jurassic Park guy with nature will find a way.
Nature does and finds different ways, which is amazing. So there's no right answer to this. There are like 25 or 30 or 40 right answers.
And the right answer can change as our understanding of it can change as we can begin to measure things differently. So think about this as if you were Captain Highliner. Why is it so hard to freeze my fish sticks? Some of them are floppy.
And why when I freeze them, they get all mushy when I unfreeze them.
Yeah.
Because ew. Okay, so these are really important questions.
Is that still a brand?
Yeah, I think it is. Okay, so let's work through this, though, from like a scientific kind of perspective, right?
Sorry, a what? That's when you put on your scientific spectacles. You're like, oh yeah, this is non-scientific, scientific.
Yay. Okay. So, we have to find the answer. Hopefully. It's kind of a cool answer,
There's
right?
some hypotheses.
Already. Amazing. Yep. Okay. So, wonderful. The best way to start is not necessarily with hypothesis, but rather an observation. Right?
So important.
Science usually starts with, what the fuck? Right?
The WTF moment.
The WTF moment.
We've been on the radio talking about a WTF
You've
moment.
coined the WTF moment.
Yeah, people were like, what about Eureka? And it's like,
no,
Eureka.
It's not Eureka. Eureka's at the end.
Eureka's like months, years later. The
Yeah.
first thing that happens is you're looking at something going, well, that's not right.
That's right.
Or as I expected. So you say,
WTF.
what's that form?
Okay, so...
Why the fracas?
Right. So you start out with an observation, something that sparks your curiosity, something that doesn't seem right or counterintuitive or whatever, right? And then you start coming up with hypotheses. Now, your hypotheses have been originally described as educated guesses. We would like you to defer that, push that aside. Hypothesis is simply a plausible explanation, a reasonable mechanism that goes that, you know, if it's this, then you write predictions. I expect to see that. Super, super simple, right? And how would you test them? And then, of course, how are you going to interpret the results, and how are you going to do your next experiment? Because very, very rarely do you just do one thing and you're like, yeah, I know the answer, right? Okay. So here are some data that we're done. We're going to kind of skip, not to the end, but we're going to skip, we're going to skip ahead and imagine that somebody did do an experiment. Okay.
That
These
phrase
guys.
is, of course, a shout out to Edgar Wright and his show Space. And
Right.
if you haven't watched it, please do.
Okay.
Skip to the end.
Here is some data. Oh my goodness, this is a really old Menti. I'm going to give you a different code
Okay, I'll put it in
in a second. What I would like you to, we'll put the code in the chat.
We'll put it in the cat.
But what I'd love for you to do while we're getting the Menti set up is to take a look at this graph and come up with two conclusions, two things, and we'll launch a short answer thing in a second that you can do in order to fill in the answers. But for now, just take a look, write two conclusions from this graph, please. And
Put on your thinking cap or your scientificals.
I will get the Menti going.
Oh, there's some good paraphrasing of WTF. Why this function? Whiskey Tango Foxtrot. What the flounder? That's good.
That's really good.
Why the flounder?
Okay, hold on just a sec.
You're out of touch.
Okay.
You're not out of time.
Okay.
Ready? Can I broadcast it?
Yes.
Here we go.
Share the Menti
The
present.
code 42910101. 42910101.
Sorry about that.
Wet the feathers. I should stop myself from reading these because
Two
you'll get me to
conclusions.
say something. Why those fellas?
Folks.
Oh.
Oh.
I was being more accusatory.
Super good. We're totally going to run out of time.
No way. Nope.
Yeah.
Nope.
Yeah,
We're doing the whole thing. You know what we'll do?
What?
We just go faster.
We'll go faster? No. No. I want to slow down on this because this stuff is really fun, and we can take the bunny stuff and do it later.
Did you just call it bunny stuff?
Bunny.
Well, we're going to have a talk about that.
Okay. Cool.
I
Is
have
that
a
okay?
I have a hair to raise.
Is that okay? If we do that?
But then we'll push the frog stuff.
We could just skip to the frog stuff.
No, the bunny stuff, the coat stuff is cool.
It's cool, yeah. but
We'll get
just
as far
cool.
as we get, and
Yeah.
we'll keep going next week.
Okay.
I love, again, so we've got like a plethora of monitors just off here, off camera.
We're
so
watching your
we
answers.
can watch your answers, and they're great.
Okay.
And it is, again, not to say, as Dr. Jacob said last time we were together, it's not that all of them are equally correct.
Yeah.
But we like the expression of them.
Yeah. Super, super interesting. Let me just kind of pop this up and you can take a look. Okay. So some of you are doing a kind of like inference kind of statement, and that's fine except for on a test. If we ask you like two conclusions based on this graph, don't like take it 12 steps forward, okay? Like two things that are being shown on this graph. But anyway, you're all very smart though, so this is good. You're going in the right direction, okay? Yeah, antifreeze proteins increases during the winter months. There's a high concentration of antifreeze proteins in the winter and the spring. Antifreeze concentration increases as outside temperature decreases. Hold that thought. The fish makes less plasma during the winter months. Super good, right? Like these are all good. Antifreeze concentrations is higher in the months where the temperatures are colder. Super. Okay. Really good. So thank you. And hopefully you can come up with two statements because there are two lines.
Yeah.
Yeah.
So I also love the fact that this is a date, like we're talking about graphing, and way back in September, we talked about good ways and bad ways to present data.
Yeah.
I like the fact that these researchers, or Fletcher and company, oriented the x-axis not to our New Year's January 1st, but to the data to express the pattern of the data best.
October, November,
so it starts seasonally in a way that you might have started off going, wait a second, how do I have to reorient myself here? And that speaks to thoughtful ways to express the data.
Super good. Yay. Okay, good. So now we have a pattern and we can start to see whether or not this holds up, right? So we have this pattern of these antifreeze proteins and these plasma freezing point. That just means like when the body's going to become ice, right? And we can see that there's a relationship, right? And you've already started to kind of hypothesize the next experiment about what's causing that relationship. What causes the antifreeze proteins to be produced in the body of the fish? And so like, what's the trigger, okay? That would be like a logical next step in this like exploration of fish that don't freeze. So let's start filling it in then from here. We have the observation that antifreeze proteins increase in the fall and decrease in the spring. Of course, that's a totally northern hemisphere bias of time because in the southern hemisphere, it's not the fall and it's not the spring. But anyway, that's a whole other thing. Question then, what is the trigger for the synthesis of the antifreeze proteins? Hypothesis. This is like you've already thrown it at us, right? In cold water marine fish, the trigger for synthesis of antifreeze proteins is, let's go and take a look over here and see if we can pull out the here we go. Here's one that's pretty much representative of some of the others. Antifreeze concentration is higher in the months when the temperatures are colder. So let's kind of run with that one, okay, since it's a popular one. Prediction, if the hypothesis is true, then what would we expect to see, right? So let's kind of draw a prediction. But before we do that, what we want you to do is to think about what do we need to do? Like what's the experimental design, right? What do we need to do in order to be able to test that prediction? So you have a bunch of fish, you have some tanks, you have some water. What is the experiment going to look like to be able to test whether temperature is the trigger for the upregulation of antifreeze proteins? So I'm going to throw a Menti at you. It's the same code, and the code is correct now on the slide. So what can we do? What does the experiment look like? Go ahead and see if you can tackle that for a minute.
We're out of touch, might be out of time. Maybe just stop it earlier when it's still legible and then we bring it back. Because they're going to get really small.
Yep. Actually, I'm kind of liking the, so a bit awkward with the word cloud format, but also quite cool. Okay. Yeah, this is cool. I like it because we've got some kind of consensus going on here. Okay, so we have changing temperature, right? That's clearly an important condition that's going to need to happen. What I'll do is, if you don't mind, we'll use the short answer from the previous question. I'll launch that right now. And so imagine that we are going to change temperature. That's critical. Now think about how. Think about how many fish. Think about tanks. Think about water. Think about like what the protocol is going to be like. And we'll let you write it all out. Here, I like this. Darn it, there's a limit. Sorry about that. Okay, so you think a little bit. So if we're going to accept changing temperature as being like the focus of our experiment, which is completely logical, control temperature, change into, you've all got this, like different water temperatures, it's all there. Let me just launch for you we'll go back to the one that I had before, you can write out like a bit more of the experimental protocol. Okay, so it says what conclusions here. What methods I will say. Okay, go ahead and it's the same code 42910101. What methods, if we're going to change temperature, what are we going to do with the fish? Nice. These are good. These are good. Good, good, good. I bet you if we put them all together, we would have a pretty good experiment. Yep. Well done. And some of you were very funny. Thank you. Okay. I like this one. Yep. These are great. This is super. Okay. Wow. Okay. You're amazing. You're all amazing. And some of you were very funny. Okay. Yeah, this is great. So, here, put multiple fish in different tanks. Why different tanks? Change the temperature over the course of X hours and watch levels fluctuate. Take blood samples to test levels of proteins. Like, amazing. Well done. Test fish from warm water and cold water, each in tanks with different temperatures. That's super interesting. Same species of fish in 5 tanks. Why 5? At intervals of temperature, and then test the levels of antifreeze protein. Slowly decrease the temperature until you start observing a production of antifreeze protein. So same fish, different temperatures. Amazing. Slowly decrease the temperature of water, measure the antifreeze proteins in the fish. Very good. Keep it at an environment with a controlled environment, but just change the temperature. Amazing. Really important. Okay. These are all really good. So how many fish? Are we talking about like 2? Are we talking about how many fish do you want to have in your experiment? And how many tanks do you want? And why is having a bunch of fish in one tank different than having one fish in a bunch of tanks each? these are questions that we grapple with, and the answer is that there are like pros and cons of both, right? What if they're a social fish, right? They kind of need to be together. If you're going to separate them, they're not going to be happy, which means you're not only going to be testing temperature, you're going to be testing happiness.
Unhappiness.
Right? But if you have a whole bunch of fish in a tank, then they're affecting each other as well. And like the environment of that tank is going to be different than the environment of another tank. And what happens if
you know why?
Sneeze into that tank and then all of your fish die?
They're peeing.
They're peeing in the tank. Oh, okay. So like these are questions. There are no right answers. There are better answers. There are controlled approaches. There are standardized methods. There are all sorts of things. How many fish? Do you want 5 fish? Do you want 20 fish? Do you want 100 fish? That's usually a money factor more than anything else. How many can you afford,
Yep.
right?
What about ages?
How old are what sex are the fish, and why do we keep on studying males only, right?
They're flashy.
They're flashy. So why these questions are important, and this is where the real creativity and your ability to shine as an individual in terms of like setting up an elegant experiment can get really quite exciting, right? And it's really important to not devise the perfect experiment, because no one can do that, but rather to understand the limitations of your experiment and do your best to kind of creatively and with a little bit of innovation get around some of the hurdles that are easier to overcome, right? And that's what's awesome about science, okay? To be able to like use your brain and solve a problem.
And don't think that this is a thing that you would study and figure out as an undergraduate and then enter the world of science and then see everybody doing things properly.
Yeah.
One of the first kind of exposures that I had to writing in the scientific environment had to do with challenging authors that had written big papers in the Proceedings of the National Academy of Science, which is like the 3rd biggest journal in the world, where they were studying the expression of an enzyme.
Oh, they must have loved you.
And they went out and they captured some frogs and they quantified the enzyme. And then they said, look, some have a big bar, some have a lot, some have a little. And they made no attempt to standardize what time of day they captured them. And this is an enzyme that responds to ultraviolet radiation. So if you did it at the end of the day, you might reasonably assume there was more of it, but they didn't. And I remember charging into this, going, well, what?
I know some things.
What the flounder?
Why have you?
Why'st thou failed?
Oh, dude, they must have loved you.
Oh, they did not.
Oh, Smith, welcome. It's so nice that you have thoughts and ideas.
They did not. Would the size of the fish matter? Yeah, probably.
Yeah, totally.
And it would matter because maybe it matters about your experiment and about the mechanism that you're trying to test. And maybe just pragmatically, it matters about how many tanks you can have, what size of tanks, what kind of number of fish you can maintain within each tank. So there's like exactly directly affecting your mechanism. Yes, it matters. Yep. And then pragmatically, how are you testing your predictions or what kind of So yeah, it matters on both sides of your if and then.
And can I add to that?
Of course.
Because it could matter biologically with the biology of the species.
Yeah, sure.
Sometimes small fish are actually freshwater fish, and then they grow up and they become saltwater fish. So if you're using small salmon, it's a totally different fish than if you're using big salmon. Or
you could be testing and asking these questions about the marine fish of the weird sex type where they are male then female.
Oh yeah, that's amazing.
Yeah.
Yes.
Wow. Yeah. So some of you just comment tank that contains all sexes and ages, but what if your fish contains all sexes and ages?
You're like checking, the fish is like filling out a form and it's going sex,
yes.
male, female, yes. Amazing.
Check out Blue Planet if you haven't seen the most recent iteration of Blue Planet.
It's so good.
Yeah.
Okay. Here's the experiment that we're going to throw at you, okay? There are problems with this. Please feel free to like rip it apart in terms of like the limitations of this. But this is what was done, okay? So using temperature, the control group had 20 fish that were held at 10 degrees Celsius. The experimental group had 20 fish that started out at 10 degrees Celsius, but the temperature was decreased over time by 1.5 degrees Celsius. Okay, so we have a control, totally stable temperature, and then we have a tank with a dropping temperature. Okay, then several measurements of the plasma proteins, and what would we expect to see? So we're going to use different colors. We're going to use a blue line and we're going to use a red line. Let's start out with the control blue line. What would we expect to see on this graph with the plasma antifreeze protein concentration over time in the control tank? So give me a blue line, please.
Super.
Yeah. That's great. I might tweak that a little bit and go, if it was the control group, they probably wouldn't be producing much antifreeze protein, so it might be a little lower at the lower end. And the thing that I like about what you've all done is that it's flat. So that's fantastic, right? Because I don't have a scale here, who knows, right? But like, just kind of keep it like near the lower end, super good. Okay, now, if we're gonna do an experimental tank, let's take a red line or a purple line, whatever you want there, a different color line that is not blue or green. And give me a experimental line. What would you expect to see over time if we decrease the temperature in the experimental group? Wonderful. Yes.
I love it.
Good. That's great.
What did they see?
So, you want to see what they saw?
I want to show them.
So this little tiny thing of like graphing out predictions, Smith and I do this with all of our students in our research group.
With our own research all of the time.
All the time.
Yep.
Right? Because it makes interpreting the results so much easier, right? It also reassures us that we have a hypothesis and a prediction, because if you can draw it, then like there's a mechanism, there's a thing there, okay?
It also helps if you're talking visually, like if you're presenting this with a group, when you have group presentations in the future and whatever, and you're all sitting in the same room and going thinking about COVID in the background, it helps keep your audience connected to, this is what we thought. So you set this up kind of like, this is what we expected, and then you can keep a little cartoon of it up in the corner like we are, and like, hello, and this is what... Now, what did they see?
That's right. So we always do that on our slides. We put the prediction on our results thing, and we go, do they match? And like a big check mark. That's like
so
cartoon science.
Since we're in that upper corner right now, just think of it like put your prediction, that's us.
Yeah, well, they can't see us right there. But
well, they will on the video.
They will on the video.
Yeah.
Okay, good. So, ready? So, what I'd love is for you to get your stamps ready, either the check mark or the X, okay? And tell me whether or not temperature is the trigger for the upregulation of antifreeze proteins. Are you ready? Here are the data. Okay, so this is an absolute what the flounder moment, okay? This is the moment...
A literal.
Yes, where some future budding scientists check out. They're like, my data are not statistically significant. There's nothing there. And a resilient and creative and curious scientist goes, wow, that's amazing. Whatever the answer is, it must be even cooler than what I was thinking. Okay, those are the pathways. You can't go in any other pathway. Okay, so thank you, Owen. You're like drawing it out here. Okay, so change the test. Nice. Thank you, Nancy. Exactly. You got to go back to the drawing board, right?
So, and this is a classic example in the literature of where a non-significant result is not synonymous with, oh, it didn't work.
Yeah.
This really worked. This is the fish talking to you saying, it's not what you thought.
Yeah.
It is not what you thought. Come back again.
Yeah. You definitely need to go and check your methods. You definitely need to, see whether or not you made a mistake. And delightfully, what should we do next? So if we go back to all the way here, we made an assumption here, right? Remember? And we totally baited you into doing it. But we made an assumption here, right? We looked at that x-axis and we said that month was a proxy for temperature. We said that as the temperature decreases, the amount of antifreeze proteins would increase. And that's why it's really important to look at graphs and actually write a conclusion without making any inferences that might be spurious, right? That might not work. What else is month a proxy? And it's not like they have a calendar and they're like, oh, it's September, right? So what else is month a proxy for? Are they getting it?
Oh, yeah.
Yay. Okay. So month is also a proxy for photoperiod.
Which you've been talking about in the daylight hours, sunlight hours.
Yeah.
Yeah. So high five yourself. Just like self five.
Good. Okay. Photoperiod.
Nice.
So here's the experiment. Okay. Control group, 20 fish. 12 hours daylight, 12 hours dark. Different tanks, different rooms. Okay. 20 fish, 10 hours in the daylight, 14 hours in the dark, 8 hours in the daylight, 16 hours in the dark, 6 hours in the daylight, 18 hours in the dark. So increasing darkness, okay? And is photoperiod a trigger for antifreeze proteins? Give me a check mark or give me an X. Yes. Good. Super. Okay. Wonderful. So we have our control group all the way on the left. We have the data presented as increasing darkness, and we have plasma antifreeze protein concentration going up, right? And we're not using the same fish. So we're not making a line or like a line graph. We're doing bars because these are different tanks, different rooms, different fish. We can do it by doing this bar chart. Okay? Super. Well done. Yay. Little story. And hopefully you've enjoyed kind of going through the thinking and the breaking down and all of that stuff to kind of simulate the thought process that goes into setting up an experiment. Now
think about this in an evolutionary and an ecological sense. So we've just, and this is why we love in this course how we are trying to link together you thinking at multiple different levels of biological scale.
You can totally run with the bunnies.
Yeah. So that you've measured something physiologically, like blood plasma. You've got fish, and you're not belly rubbing. You're extracting blood, and people are like, "What do you do?" It's like, "I measure blood," but you're thinking about it in an ecological and an evolutionary context. And now your experiment, the fish have told you, it's not the temperature as you thought; it's the photoperiod. So then think about it evolutionarily. Why? Why is this a better trigger? Or why is this a trigger? Why is this the evident trigger?
Right. It's, so buddies like adapting to cold, but using photoperiod instead of cold. Just like we were using month instead of photoperiod. These proxy variables, like which one is the meaningful one? How can it evolve? What is the context under which this can evolve? And I hope some of you have just gone, oh my goodness, that's evolution and ecology and physiology all locked into
together.
One. Look at us.
Yeah, so there's some thoughts in here. One of them one of the first ones I saw, M. Corneo 2, more consistent. So what are you thinking about when you say more consistent? How is photoperiod more consistent? Because you're on the follow that.
Follow it. Yeah.
Photoperiod has less fluctuation. Temperature can fluctuate. Photoperiod changes before temp. Yep.
Yeah. This is such a smart group.
Yeah, this
well
is
done.
awesome.
Okay, yeah. Yes, temperature kind of thing, right? There's temperature and there's climate, and it's all kind of like weather and climate all kind of mixed up, right? And so over time, like if you integrate temperature changes like weekly, yes, you get a nice straight line. But daily, you get this daylight, nighttime fluctuation in temperature, and then also from day to day, sometimes it's colder, sometimes it's warmer, and it's all kind of messed up. And if you've got some kind of physiological trigger that's
microhabitats can change it.
Right?
Yeah, so micro time, microhabitats, all of these things.
That's right.
There's an honesty to the photo period.
Yeah. Exactly. Oh, an honesty.
There's an honesty to it.
A reliability, right? And then things are messed up. So go away.
Yeah,
Take it with the buddies.
so things are messed up. How might things be messed up? Well, it's kind of how we're living through the world right now. So we're going to talk about hares, not about bunnies. Look at the length of those ears. These are clearly hares. So nice long ears. I might throw some, there's going to be some examples that involve snowshoe hares, which are kind of boreal mountainous things, big wide range, or Arctic hares, which is, as you might expect, much higher up in latitude. But both of those species, they're in the genus Lepus, which is easy to remember because they leap. What do? Us, we leap. We're Lepus. They exhibit coat changes. So they have a summer color, a brown cryptic color, and then a white color.
Yay! So pretty.
Now these are actually the Arctic ones, and you can tell that because of, well, what's in the background, but also that the length is actually of their ears is slightly shorter, which is another kind of an Arctic adaptation. Like, don't have big elephant ears because you'll lose heat. So what might happen? Why might that be? So many Arctic and montane animals actually seasonally change their coats. So part of that has to do with camouflage, that if you're a nice dark brown hare in the middle of the wintertime in Nunavut, that probably means that you're going to be a very easy to spot meal for something else. Think lynx and hare. And the lynx would be like, that's easy, I'm eating here tonight. The other reason that white guard hares change seasonally is that they often contain the ratio of kind of pigment to air changes, and it means that they can potentially be more insulative. So two different periods on this graph. On the right hand side, we have Yukon hares measured in characteristics of hair, pelage or hair. I was like, I think I just said hair. No, it's like, it's hair. So from the fall to the winter, kind of going backwards in time in this graph, and if you click on the graph in the PDF later on, you can go and take a look at the original data. The length of the guard hair increases. The density of the guard hair increases. The density of the downy hair increases. The length of the downy hair doesn't really increase because then it wouldn't be downy anymore. So that's the one on the bottom left that kind of stays the same. But you can see all of these investments that hares make to their hair associated with a change in, it feels like intuitively temperature, but quite honestly, with photoperiod. Now, why might that be? Oh, so the first then, you do the Nunavut one.
And let me just throw this at you. We're going to go over 2 minutes, but I hope you don't mind because it's cool. Okay. The other thing is, great. So hares change fur color in order to look more like their backgrounds, except if you're like a super duper Arctic hare. If you're like an Arctic hare, like a mid Arctic hare, then you totally do that. So these two photos were taken in the summer months when Arctic hares should be brown. It turns out that in the super duper north, the Arctic hare stays white the whole time. It doesn't change color. It does get a summer coat that is thinner than the winter coat.
So the downy hair.
Right, but it doesn't change color. What explains that? And then like you start to unpack this stuff and it gets more and more awesome, and then you have like a whole career lifetime of like amazing and interesting research. Here's the line, below which we have hares that change color, above which we have hares that stay white the whole time.
And some of the why you might be asking, and some of you asking, so it's just for camouflage, blah, blah, blah. So it is for camouflage, but the fact that this exists, I was writing in the chat, talking with somebody earlier about, well, why might this happen with a flounder example? And it has to do with the fact that any of these strategies are energetically costly.
Yeah.
If you're doing this, it means you're not doing something else. You're not feeding, you're not growing, you're not producing babies or producing gametes to make babies in the future. So the fact that if you would opt out of it, that suggests there's a benefit.
There's a benefit to opting out if up here your summer is so short that changing your color doesn't really benefit you compared to the energy that you would be saving to do something else. So yes, in the high Arctic, they do stand out, like in this photo,
Yep.
for a few weeks. But it's the lower Arctic that has a longer summer of this like gray, dark brown background, where having that color change really does benefit you because it's a matter of months now. That's kind of the idea, right? This like cost benefit trade-off kind of balance, right?
But think, just in this last minute, think about the fact that this is possible because of the honesty of photoperiod, because of the tilt of the Earth away or towards the sun. But what's changing now? For the hare and for all of these other 9 mammal species that do this kind of thing, largely across latitude, but also across elevation with, who is it? The weasel that extends down along the...
So what's changing? The honesty of the trigger works. This totally works, right? And
we've known it for a while.
We've known it for a while. Okay. This is somebody in 1942 saying this. So the problem with this has to do with what's going on right now with the climate crisis. The problem is that it's no longer an honest signal of the actual temperature. The problem is that the super duper northern hare is now going through a summer that is so much longer than it used to be 20, 30, 40, or 50, 60 years ago. And it's becoming more and more costly to have the white color the entire year round, right? Similarly, for the one that's further south, there's a mismatch in the timing that the trigger has evolved to and the actual stressor or the actual thing that it's adapting to.
So this mismatch could happen in the fall or the spring, and it results in then hares that are brown when things are white, or hares that are white when the things are still brown.
Yep. So staying warm, keeping cool, really does depend on the ecological context, on the abiotic conditions, on the changing abiotic conditions. And there can be these evolutionary selections that if it's slow enough anyway, you might see changes or adaptations in the future. So just kind of bringing it all the way around back to the evolution unit when we think about this. So we'd love for you to think about it. Here's your homework. From an ecological, physiological, evolutionary perspective, what are the consequences of all of this if the Arctic hare are changing their colors in some regions and not in the context of climate crisis triggered by photoperiod? It's a lot.
Yeah.
Take care.
But that's all right. The world is a lot right now. Take care of each other. Be kind.